Today, an FDA approval of a biosimilar, promising findings in multiple myeloma and glioblastoma multiforme, disappointing results in pancreatic cancer, and a nomination for the next FDA commissioner. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved the pegfilgrastim biosimilar LAEP2006, known by the trade name Xtenzo, as a treatment to decrease the incidence of infection in patients with non-myeloid malignancies receiving myelosuppressive anti-cancer therapy that is linked with a clinically significant incidence of febrile neutropenia. The approval is based on analytical, preclinical, and clinical studies, including data from the pivotal three-way pharmacokinetics and pharmacodynamics LAEP06104 trial. The study compared the pegfilgrastim biosimilar, which is developed by Sandoz, with U.S.-sourced reference pegfilgrastim, the pegfilgrastim biosimilar with European Union-sourced reference pegfilgrastim, and U.S. with EU-sourced reference pegfilgrastim. Results show that PK and PD similarity were demonstrated in all three comparisons, and there were no clinically meaningful differences observed in safety and immunogenicity among the groups. In November 2018, the European Commission approved LAEP2006 as a treatment to reduce the duration of neutropenia and incidence of febrile neutropenia that is associated with anti-cancer chemotherapy. In multiple myeloma, ixazomib as a first-line maintenance therapy significantly improved progression-free survival compared with placebo in adult patients who have not undergone stem cell transplantation, meeting the primary endpoint of the Phase 3 Tourmaline MM4 study. The trial comprised the concept of switch maintenance therapy and enrolled patients who had completed 6 to 12 months of initial therapy and achieved a partial response. The top-line findings also showed that ixazomib's safety profile in this setting was consistent with previously reported data of the proteasome inhibitor, and no new safety signals were reported. Full findings will be submitted for presentation at an upcoming medical meeting. The FDA initially approved ixazomib in 2015 for use in combination with lenalidomide and dexamethasone as a treatment for patients with multiple myeloma who have received at least one prior therapy. Ixazomib also showed promising activity in the maintenance setting. Results of the Phase 3 Tourmaline MM3 trial showed that 2-year maintenance treatment with ixazomib led to a 39% improvement in PFS compared with placebo in patients with newly diagnosed disease who achieved a partial response to induction treatment with a proteasome inhibitor and/or an immunomodulatory agent following autologous stem cell transplant. In interim findings from a Phase 2 trial, the combination of INO-5401 and INO-9012 with cemiplimab plus chemoradiation demonstrated promising 6-month progression-free survival rates in patients with newly diagnosed glioblastoma multiforme that is MGMT methylated or unmethylated. Results of the 52-patient study showed that 80% of MGMT gene promoter methylated patients and 75% of MGMT unmethylated patients were progression-free at 6 months, measured from the time of their first dose. These data are said to exceed historical standard of care results, stated Inovio Pharmaceuticals, the developer of INO-5401 and INO-9012. Detailed results of the study were presented as part of a late-breaking abstract during the 2019 SITC annual meeting, which includes supportive safety, tolerability, and immunogenicity data. The combination also demonstrated an acceptable safety profile that was consistent with that of cemiplimab, as well as INO-5401 and INO-9012. Final data from the Phase 2 trial are expected in the fourth quarter of 2020. In pancreatic cancer, PEG-4 alpha, known as PEG-PH20, in combination with gemcitabine and nab-paclitaxel, did not show an improvement in overall survival compared with gemcitabine and nab-paclitaxel alone in patients with metastatic disease, missing the primary endpoint of the Phase 3 HALO-109-301 trial. Results show that the addition of PEG-PH20 led to a median OS of 11.2 months compared with 11.5 months in those who received gemcitabine and nab-paclitaxel alone. There was a higher response rate in the PEG-PH20 arm, but it did not translate into an improvement in progression-free survival, OS, or duration of response. Due to these findings, Halozyme, the manufacturer of PEG-PH20, stated in a press release that it intends to halt development activities for PEG-PH20 and will implement an organizational restructuring. Prior data demonstrated that PEG-PH20 showed a benefit in patients with metastatic hyaluron-high pancreatic cancer. In the phase 2 HALO 109202 study, the objective response rate in patients with HA-high tumors was 45% with PEG-PH20 plus nab-paclitaxel and gemcitabine versus 31% with nab-paclitaxel and gemcitabine alone in patients with untreated metastatic pancreatic ductal adenocarcinoma. President Donald Trump is nominating Stephen M. Hahn, MD, FASTRO, as the next commissioner of the FDA. Hahn is currently serving as the University of Texas MD Anderson Cancer Center's chief medical executive and was reportedly the preferred choice of Department of Health and Human Services Secretary Alex Azar. If the nomination is confirmed by the Senate, the radiation oncologist will serve as a successor to Scott Gottlieb, MD, who resigned from the role in March 2019. This would be Hahn's first venture into public service in over 2 decades. Acting Commissioner Ned Sharpless, MD, who was appointed to the position in April 2019, is expected to remain in the position throughout the confirmation hearings. Sharpless is expected to return to his role at the National Cancer Institute if the nomination passes through the Senate. To meet the terms of the Federal Vacancies Reform Act, the Trump administration has temporarily named the current HHS Assistant Secretary of Health, Brett P. Garor, MD, as the interim FDA chief until Hahn is appointed into his position. Throughout his career, Hahn has also served as the chief of the NCI's Prostate Cancer Clinic, the chairman of the Radiation Oncology Department at the University of Pennsylvania Medical School, and is the author of over 200 peer-reviewed journal articles and publications. This week, we sat down with Dr. David Hong of the University of Texas MD Anderson Cancer Center to discuss future challenges and evolutions with TRK inhibition.
What are the ongoing challenges for TRK inhibitors? One, I think the main challenge is really to help, how do we identify these patients? Because these patients do have incredible benefit from larotrectinib and other TRK inhibitors. So that remains a challenge, right? Because I would argue at this point, even in the United States, the vast majority of patients with metastatic cancer of some sort do not get tested with NGS or other standard platforms. I heard a statistic out there that lung cancer patients, only about 40% of patients actually get even tested for standard mutations such as EGFR or ALK-ROS. And so I think that remains a major challenge, right? How do we ensure that patients increasingly get this NGS testing? All patients at some point, or most patients at some point, will develop some type of resistance to any therapy, especially in the metastatic setting. To date, this drug has had incredible durability, and as I shared, median progression-free survival is 25.8 months. These patients remain on study for years, sometimes. The hope is that many of these patients will remain on study for years, but as we know from past experiences is that most patients who go on a small molecule inhibitor will eventually develop resistance. And our colleagues around the world, and Sloan Kettering and myself, are already developing the next generation of TRK inhibitors. There are other TRK inhibitors that have already emerged that target specifically mutations that may arise from resistant phenotypes such as solvent front mutations, gatekeeper mutations. And so that will be the next kind of generation of TRK inhibitors. I'm sure there will be other mechanisms of resistance that we discover as we learn more about this disease and about this pathway. And so, but I think what's hopeful and what's exciting is that the chemistry has gotten better, the biology has become more understood, right? And we are moving very quickly to implement with our colleagues at the FDA to get new drugs to our patients. So I think it's really a hopeful time in oncology.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.